what I want to talk to you about today is two conceptual introductions, how to think about cell identity and cell communication, and then specifically how we can analyze spatial transcriptomics data to some extent to answer these more conceptual problems. So the plan for today will be, and I should also say a few words about myself. I just got PhD. I did my PhD in Bayraktar, Steglia, Teichmann groups, and also working closely on some project parts with parts groups at Wellcome Sanger Institute. What I will show you today is the focus of my PhD work, and you will see all about it. Before PhD, I did a lot of internships actually in molecular cellular biology. So I have background in biology, but now, as some colleagues say, I'm an honorary physicist and statistician. I didn't study at the university to get those things, but I learned how to do them. That's short.
Where did you get your bachelor and master?
In Taras Shevchenko University of Kiev. I'm not particularly proud of that stage of my career. But yes, that's where it happened. So the plan for today is to discuss how we can formalize what cell identity means, then do the same but for cell communication, and briefly discuss spatial architecture as an additional aspect of why we need spatial transcriptomics data, and then I will explain you the tools that we developed, as well as give 3 examples of how it can be used to study cells and tissues. Most of the time we'll be here. So first, thinking about cell identity, this is one of the kind of core interests for me, and took a bulk of the PhD work. What we want to do and what we want to formulate is a mechanistic model of cell identity. How do we do this? We need a model that represents cell state. Specifically, it represents the abundance of transcription factors, kind of proteins that regulate transcription, as well as certain information about cell communication. Then the model needs to represent the mechanistic steps that are involved in how those proteins regulate what happens to the genome. And what happens to the genome, a lot of things happen to the genome, but the things that we can easily profile across hundreds of cell types are DNA accessibility and RNA abundance. So what we want to do is we want to have a model that looks at transcription factor abundance as well as abundances of signal and receptor molecules, and processes that in a mechanistically motivated way to give us cell phenotypes. And we need, and just formulating computational model isn't enough, because we need to show that model highly comprehensive cell atlases of cell identity. So here, for example, is our work between Bayraktar, Teichmann, and Mus Hanifa groups, where Mus Hanifa group can obtain human embryos at stages of middle to late organogenesis, where we get the entire embryo with all of its developing organs. And what this allows us to do is it allows us to generate single cell RNA-seq, ATAC-seq from exactly the same cell in highly consistent way, such that we can compare the cells across the tissue. And this is just the UMAP representation of lots of cell states, mainly showing that this is, if you look at the entire embryo, it's super heterogeneous, and I will show you some examples of where these atlases are actually super relevant. and color shows these anatomical sections. We need this comprehensiveness to show models what happened, what kind of cell identities exist, and the model needs to be sufficiently kind of complex to explain all of this stuff. You can also ask why mechanistic models of cell identity, and I think the answer to this is that we need minimum required complexity models. You can think of it as on one end, you have simplistic models like a linear model. We can fit linear model to anything and say that it fits, analyze the model, but that would be a simplistic model for complex processes such as gene regulation and cell communication. On the other hand, you can have an overcomplicated model. Like in physics and chemistry case, I guess you can say that, well, we will fit we will use quantum simulations to reflect the properties of atoms and do everything at the most detailed possible level, but this is also not correct. What we need to do is we need to find the sweet spot of what are the most important parts of the process that we are trying to describe, gene regulation in this case, what can we measure experimentally, and how these things connect, and we need to find a model in the sweet spot. And you can realistically only do this if you go through the process of doing mechanistic work as opposed to deep learning, as opposed to kind of trying to black box everything. And another reason is that mechanistic knowledge allows you to formulate a kind of more efficient model, both more interpretable as well as that learns better. And also to evaluate if these models work, you need to understand some mechanism to be able to kind of create benchmarks that dissect what needs to be dissected. And on top of these more dry reasons to have a mechanistic model of cell identity, we need that to answer basic questions around how cell type specificity of gene regulation arises from underlying components, as well as kind of these days people look a lot into plasticity, especially in cancer contexts, or I mean in development obviously, but also in cancer contexts. And this is generally done with heuristic models that don't really represent the basis for plasticity, and these types of models can be that basis. And I will show you one example of where we got so far. And finally, if we want to create a cell engineering tool that allows you, kind of computational cell engineering pipelines that allows you to predict how you need to perturb cells to direct, let's say, certain disease state towards more functional state, we again need models of mechanistic models, because we need to be able to perturb things as they happen. Yeah.
Can I ask a question? Like asking questions is allowed during your presentation?
Yeah. So I was thinking that maybe we should ask questions at the end of each section, just to not run out of time before we see the more practical part. Okay. Ooh, I see. It's got stuck. So there are. I also said that we need a single model to explain all of that, all of those cell states. In what we need to aim for is a whole organism, the Atlas whole organism model. And the reason we need to do that is because DNA sequence is shared by all of those cells, but with some exceptions like T cell receptor, B cell receptor editing. DNA is shared by all cells, and this means that what is shared is both the sequence of the regulatory elements as well as the sequences of regulatory proteins such as transcription factors. And on top of this, I guess this doesn't always work. On top of this, we have transcription factor and cell communication environment reuse across different contexts and similar TF-DNA recognition preferences. So now I will show you what I mean by those 2 points. One of my favorite examples of TF reuse is DLX5. DLX5 is a transcription factor known to regulate neuronal identity. And here in this atlas, I'm highlighting nervous system with these 2 cells of the nervous system with these 2 outlines. And you can see some DLX5 detection in cells of the nervous system, which likely correspond to cortical inhibitory neurons in this case, and peripheral nervous system cells. But surprisingly, we also see DLX5 detection in many other cell populations, including stromal cells of the gut, hepatocytes, and some other populations. And what this suggests is that even transcription factors that the literature says are regulating cell type X, and that you can even use in vitro to induce formation of that cell type from stem cells, even those transcription factors are reused in multiple contexts, and we actually need to disentangle that. So what that transcription factor reuse means is that to deal with it, we need to explicitly model abundance of transcription factors. Initially, just RNA abundance as detected using single-cell transcriptomics. And second is that because we now have to model abundance differences, we need to associate those abundances with what kind of DNA sequences transcription factors bind. So this is often represented as these motifs, and it's derived from in vitro experiments where people mix in vitro produced protein with naked DNA, with random nucleotide naked DNA, and then perform rounds of enrichment to figure out which kind of sequences are bound. And you can look at this paper for more details on how you should do this. I think that paper has some very good ideas. So on top of but we all know that RNA is not protein, and we also know that transcription factors don't work alone, that they are we don't know the full extent. We certainly know that there are families of transcription factors that are dependent on the activity of these signaling pathways. For example, SMAD family, which is regulated by BMP pathway. So we need to introduce additional terms in the model that describe the activation state. 2 types of activation state. First is chromatin-associated abundance of TFs. You can think of it as transcription factors with necessary post-translational modifications and nuclear localizations that would enable them to work in the nucleus and to produce effects on our phenotypes. And the second is that transcription factors may be activated not just to do nothing versus do something. They can also have active repression or active activation state due to how they use cofactors. So we also need to represent that in the model. So then the second big point I mentioned is that transcription factors often share almost identical DNA binding preferences. So here is an example of these motifs for a certain family called EBOX family. As you can see, kind of what I didn't mention, I guess, is that what this logos represent is that the higher the letter, the more preferred that nucleotide is. And if you see several nucleotides kind of overlapping, this means that both of them have some amount of influence. So like CA here, if you can see anything. But overall, the most interesting point here is that DNA binding domain, kind of part of the TF that binds DNA, is conserved between proteins of one such family, and that proteins that are known to regulate very different fates, including opposing fates of oligodendrocyte development, which are kind of supporting cells in the brain and peripheral nervous. Well, actually, in the brain they are called oligodendrocytes, and neuronal lineage. So you have transcription factors that specify distinct lineages, yet independently they bind identical sequences. So we need to resolve this complexity. And to do this, we need to, and actually the second complexity we need to resolve is multiple mechanisms of cooperativity. So, yeah, I will just say that we can discuss this later if you want to know more. So to resolve the identical motifs, we need to use transcription factor cooperativity models of how possible binding sites interact with each other, as well as to normalize to say that, well, if we have two TFs that are both present in a cell, they cannot, they have to compete for the same binding site. And with all of these ideas in mind, we can now put together a model that goes from left to right and describes how we go from transcription factor RNA abundance, information about signaling molecules, from reading DNA sequence to prediction of the DNA accessibility phenotype. At the moment, we struggle to predict RNA abundance, and by we, I don't mean just kind of our work in this project, but in general, the field. It seems that we are missing something about how to do it correctly. So yes, it's a very complex slide, a lot of things happening. I'm happy to talk about this later. Here I just wanted to say that if you fit all of these theoretical parts together, you can formulate a mathematical model, you can formulate a code that allows you to specify all of these steps. You can train this model on the atlases that I showed, and you can analyze what happens. So, just briefly, does it work? Now, here is an example of cell type distributions for two arbitrary DNA regions in the genome. As you can see in the top example, prediction on the left, observed data, observed accessibility, DNA accessibility signal on the right. As you can see here, the model learned how to correctly explain accessibility of that DNA region from transcription factor abundance and activation states, as well as directly DNA sequence. Whereas here, DNA accessibility is still very specific, but the model doesn't predict as specifically. So kind of there is some way to go, but given that it sometimes works, can we use it? And here is an example about that plasticity point that I made earlier. The idea behind plasticity in development is that if we look at progenitor cells, these progenitor cells already have certain transcription factors, and what happens when those progenitor cells gain different fates is that depending on their environment, the progenitor cells get different signals, and that leads to activation of one of several of these TFs. And we wanted to test this with this model, so we trained the model on actually on a mouse embryogenesis atlas, and what we tried simulating here is taking progenitor cells called neuromesoderm progenitors, and I've highlighted in black, and testing whether transcription factors which are already present in those cells can be activated in ways that give you either mesoderm on top or spinal cord at the bottom. And we see some encouraging results, but we all, overall what this can suggest is that we can simulate cell state, and because we have this more mechanistically motivated model, we can analyze things like transcription factor activation, not just is this transcription factor detected in cells or not. Yeah. So let's say this is how we approach modeling cell identity, kind of how we go from concepts about cell identity that many of you know to the exact computational models, and hopefully gives you an idea of where we start in this space conceptually. And if you have any questions about this section, let's discuss them now. Yeah, I want some questions. Then.
Yeah, I just wrote, but that's that it's regarding your beginning of the lecture.
Yeah, sure.
So I had a question regarding you are saying like you need mechanistic model, and therefore you assume that you know the diversity of models, and you pick the mechanistic one. Could you please explain what do you like? What do you mean, say, a mechanistic, and then what other models also are available, but we don't pick them?
Yeah, that's a good point. I mainly think about distinction between black box models, think of any deep learning architecture, and models where the architecture is based on what we know about biological process. So in this case, we want the architecture based on what we know about how transcription factors work, that they are influenced by cell communication, that they cooperate when binding to DNA, as opposed to just taking a deep neural network, giving it the same inputs and hoping that it does what you want. You can also have, let's say, you can somewhat think of the black box deep learning as an overcomplicated model. And what we do here is we narrow down the complexity using this prior knowledge. And people also call this inductive biases when designing the architectures. But yeah, on the other end, you can fit simplistic. I mean, it's not really. I don't want to say that this is a very concrete gradation, but a helpful way to think about this is that you can fit statistical models. You can know that. Well, you want to predict transcription factor. You want to predict RNA abundance from transcription factor abundance. So let's formulate a linear model with weights for every transcription factor and every gene. And this is what is done by kind of GRN model family. Hopefully this answers that.
For the model, when you are comparing with the experimental data, what metric are you using for making the comparison, and how do you select the parameters?
Yeah, that's a very good question as well. Yeah, we actually came up. This is not just a very good question. It's very important. It's very important to get this right. So. I can repeat the question. So the question is, how do we validate these models? How do we train them? And how, which metrics we use to compare them to observed data? And a very important point about training these models is that we want to show them, and if this is you can think of it as gene expression or DNA accessibility metrics with cells here, DNA regions across chromosomes here. What we want to do is we want to stratify both cell types and cells and chromosomes into training validation test sets and show the model only a subset of all chromosomes and all cells and all cell types. And this works and then evaluate the model on new cell types, or as other people do when they apply deep learning models to this problem, the same cells and cell types, but new chromosomes. And what this does, it tests whether the model learns mechanistic knowledge. So whether, if you give it new sequences, new chromosomes that it has never seen before, can it make correct predictions? And this is a big difference between how you do a more mechanistic sequence-based models, DNA sequence-based models, and GRN-style models where you just fit statistical relationships. And how do we evaluate? So you can evaluate in multiple dimensions. The thing that we are most interested in is describing the differences across cells. And at the moment, we just use Pearson correlation between normalized observed data and normalized predictions. But yeah, cool. Any other questions about cell identity or this work? Okay, then let's go to the next step. What I mentioned in this section is that yes, transcription factors don't work alone and that they can influence transcription factor activity in this way. But now we need to talk much more deeply about what cell communication actually means. And how we can relate the special transcriptomics data sets to what we know about these processes. So first, let's discuss kind of what are the entities involved in the cell communication problem and what evidence we have for them. And the first thing we should look at is similarly to how we looked at transcription factor case is RNA abundance, because this is what is measured in single cell transcriptomics and this is what we can do. But this is also the first level, and it's about detecting RNA of receptors as well as kind of signaling molecules, and the signaling molecules can be both secreted as well as transmembrane. And this you can just directly observe in single cell atlases. However, again, RNA is not protein. We ideally also need, not ideally, I mean, we need to model this properly, we need to know protein abundance of exactly the same molecules. And you can measure this to some extent with technologies such as CytSeq and also in situ protein imaging. So both of these methods rely on antibodies. Well, in situ protein imaging can also be done in different ways, but both of these methods rely on antibodies to detect this target proteins. And then on top of this, even if you observe a protein in a cell, it doesn't really mean that it's in the functional state. For example, signals need to be secreted, receptors need to be on the cell surface. And you can only get this evidence if you do in situ protein imaging. Like if you actually stain proteins in the tissue and image them at high resolution, see what's happening. So these three levels describe you what happens to cells. But cell communication involves distinct proteins, so we also need information about those proteins directly. And third thing we need to know is the affinity between these signaling molecules and their receptors. And you can only get this from protein binding experiments. There is a range of technologies. Discussing them is a bit out of the scope of the current talk, but there are technologies that allow you to determine whether transcription factors interact and at what intensity, at what affinities. And we can use those technologies to determine what binds what. Crucially, in this case, what the literature is finding is that you can have very drastic changes in affinity as you move across protein families. So if you compare BMP protein family to VNT protein families that are kind of major cell communication families that are used in development, they would have very distinct binding preferences. But within one family, you have a gradient of affinities. So then as next level, what we need to know is not just whether the signal was secreted from source cells, but actually if it's around target cells, and what is the concentration around target cells. And you can try to estimate this from spatial RNA sequencing, as I will mention later. But in general, you have to directly measure it by imaging proteins. And now if we put the affinities and the concentration of receptors around target cells together, we can get the occupancy of receptors by signals, and this is what cells actually experience. And a cell a cell of any particular cell type has tens of receptors or even hundreds of receptors on their surface that sense different molecules and can induce different downstream consequences. So let's now think a bit about what happens after a signal is bound to the receptor. And similarly to how we went through thinking about this problem for cell identity, let's think about kind of formulating a model for this. So on one end, we have kind of mechanistic state variables, where state variable just means that you characterize the state of the cell. It describes something which is not in the rules of how the model operates. You have state variables. Let's say we understand what they are in this case. They are abundance of signals and receptors. Maybe we can directly measure just RNA level. But what is harder to formulate is what are the phenotypes? What is this model supposed to explain? In single cell case, it was relatively easy. We measured RNA abundance, we measured DNA accessibility, so let's just predict that. In this case, it's harder. And it's harder because there are multiple things that can happen when cells are exposed to signals. I would encourage all of you to read this TG biology review by Miri Adler and Ruslan Medjetov. It's quite informative in outlining a lot of the points we are talking about today, but specifically on TG architecture and cell communication. And there is a number of actions that cells can take. They can either just stay in whatever state they currently are, not die. They can die, they can replicate, they can change state, or they can change location, like migrate or get recruited. And the problem is that if we look at the spatial data, if we don't directly design measurements to detect each of these types of actions, we only observe collocation. And it's somewhat easier, let's say, if we don't. So what this means is that we actually, to distinguish these mechanisms, we actually have to design either models that allow you to separate them or direct measurements. And let's think about the model. So what we want, again citing this review, is we want to describe this type of instructive relationship between cells where source cells send signals to effector cells, and then effector cells change something depending on that signal. And based on what we just discussed about transcription factors, we can disentangle this process by introducing the mechanistic model of cell identities that we just discussed. So we can say that transcription factors are present in source cells that are exposed to signals, not source cells, in starting cells that are exposed to signals. And depending on signal exposure, they get different transcription factor activities, and we can try to fit these models. And this is a more complex case, which is the focus of our ongoing work, as well as, I think, the future for the field. However, what we have now in practice, kind of, as you may have discussed, kind of heard about during earlier presentations in this course, is that you can use databases such as CellPhoneDB that tell you, they don't really tell you signal receptor affinity, they just tell you which receptors bind which ligands in a binary way. But you can combine these databases with information about signal molecules and receptor molecules in source and target cells. And what this tells you, if I don't, what's important to say is that it gives you an important layer of information. It tells you which cells have the potential to interact, but you don't know whether they interact, and you also don't know what is the consequence of that interaction. If you just have a single cell atlas with this evidence and a database with this evidence. And what we want to do is we want to get at, do they actually not just have the potential to interact, but are they in the same spatial space? And we can get that by doing spatial profiling. However, the challenge is that in the spatial data, we also have some constraints. So specifically what we can see in the spatial data is where the target cells are, their receptor RNA abundance, where the source cells are and their signal RNA abundance. But what we don't have is this image. We don't really know whether those signals reach target cells, and we also don't know the receptor occupancy. So there are lots of limitations on what you can see with these data sets. However, this could still be a valuable information. So to end this section is that, what we want to be able to look at cell communication effects on cell identity is a detailed model that quantifies all of the steps in the correct way and also leverages mechanistic models of cell identity. But what we have at the moment is just a way to measure signal RNA abundance around target cells. So hopefully all of that makes sense. Now let's discuss if there are any questions about cell communication part.
I have a question. You mentioned that vision is used for spatial transcriptomics. So my question is, it operates on spots, so it's not single cell resolution. How is that resolved?
Yeah, this is a very important point which we will discuss shortly when we come to the technology discussion. But yes, please all take note of that question. Don't treat spatial transcriptomics as cells. Any other questions about cell communication?
I had a question about how you are considering signal RNA abundance and receptor RNA abundance. I was wondering how, in terms of proteins in the extracellular matrix, the source cells need not always be actively transcribing in order to produce a signal. So how would you reckon with that?
These are all the important layers, and the question is, how do we deal with extracellular matrix? At the moment, we chose not to deal with it much. One way you can deal with it is that you can treat extracellular matrix proteins as secreted proteins. They are somewhat conceptually similar because they are secreted from cells, but they are different in that they form stable mechanical structures, and you can also get cell communication via mechanical signals rather than protein affinity specificity recognition. Yeah, at the moment we are not dealing with it, but that's an important thing to consider. Again, what I think I need to highlight is that when we are looking at spatial transcriptomics, not protein measurements, we are looking at transcriptomics. So what we see is this picture, and you have to kind of overlay that knowledge, like your point about extracellular matrix, onto this picture, because this is a picture that you observe. So you need to think about how in the source cells, if you detect RNA abundance of extracellular matrix in source cells, this suggests you kind of a rough place of where those cells might be producing the extracellular structure of the tissue. But you kind of have to think about this picture that you observe RNA and in this type of manner.
I think I'm missing a part. So basically, when you do cell communication analysis and single-cell RNA sequencing, then you see presence of mRNA or some receptor on one cell type, then ligand on another cell type, and then you just assume that they may communicate. So basically, spatially, if you have this information, and now you know that they are close to each other, I mean these cells, this receptor and ligand, then you just assume by default that they're communicating. And what is the question here? Because basically, if you have spatial transcriptomic, and then you know that the cells, they match in receptor and ligand, and they are in close proximity, then apparently they are communicating.
So it doesn't prove that they are communicating. It just tells. So if you just have single cell atlas, you know that cells have the potential to interact. If you add this special dimension, you indeed know that which cells are in proximity of each other, and this can help you guess at this picture. So it can Yeah, actually. What you want to know is a signal concentration around target cells. And what spatial transcriptomics tells you is that cells that are secreting signals are close to cells that are recepting that signal. So it's possible that signal from these cells reaches target cells. But you still don't fully know whether that happened. So you reduced the space of potential interactions, but you still have a lot of questions. And one of the main questions is that you don't really know what is the consequence of that interaction. So you don't really know what happened in response to that interaction. You just see collocation. So there is a lot of entangled phenotypes.
Then what you need, basically, you need to kind of to film. I mean, you need to have a movie during some time after you target the cell, and then see how RNA expression changes during the time, right?
Or not just RNA expression. I mean, what would be the ideal experiment is something that Ron Germain group does on their work on immune responses. They have mouse models where they have visualized different cell types using different fluorescent proteins, and they can perturb cells in vivo inside mouse. For example, they can kill cells and see how various cells of the immune system respond to that, how they get into the tissue area. And if you then on top of that visualize the right proteins, then you would be able to see this layer. And in our work, what we are trying to do is we are trying to predict this layer by using a combination of information that you get from single-cell atlas and the RNA abundances in space. But this is obviously limited. And you also still have the problem of disentangling phenotypes. You have the problem of disentangling phenotypes, and we want to say that we can disentangle cell state change from all of the other options because we have a very constrained mechanistic model of cell state. And it's constrained by DNA sequence, so not everything. So kind of the problem here is that a lot of things correlate with a lot of things, and one of the references that I would recommend you to read is it's fantastic work from Fabian Theis group by David Fisher. Method is called NSAM. I don't remember how the paper is called. They discuss a lot of these computational modeling issues, including how you have to deconfound potential consequences.
Could you please repeat the name?
David Fisher and NCEM. I can send it on Slack. Cool. So yeah, cell communication is interesting, and this is one of the things that make me go to work. But I don't want to give the wrong impression that the only reason to look at spatial architecture of tissues is cell communication. You also have other aspects that are related purely to tissue architecture. And here I want to cite again the same review from Ruslan Mejitov. Most tissues are not just a random mix of cells. They are highly structurally, their architecture is spatially arranged. So often you have an epithelial layer on top with stromal layer at the bottom, and this stromal layer needs to contain various cell types to kind of create a functional barrier. So if we talk about tissues such as skin and gut and lung, kind of lung airways. And what they propose in the review is that rather than thinking about cell types and where they are, we need to think about cell relations. And the two cell relations, which I think are critical for thinking about tissue architecture, are in functional dependencies. One is complementarity, so maybe to do some function, you actually need both cell types. And these cell types need to be spatially arranged in the right way. And one of my favorite examples of these mechanisms is liver zonation, where the way liver is structured is that you have gradients of cell identities along the path through which blood flows. and cells earlier in that gradient can do some metabolic functions that are necessary for later cells in that gradient to do their metabolic functions. So you can have this very strong complementarity where things just don't work if you don't have this particular architecture. And another example is supportive relations, where neurons are the main cell type of the brain, but they need other cell types in the tissue, such as astrocytes and oligodendrocytes, to work. And astrocyte idea and oligodendrocyte idea doesn't really make sense without a neuron. But depending on which neurons are in which area especially, those other supporting cell types also need to adapt. So the space of questions around spatial data is much greater than cell communication. And with this in mind, I think we can jump to the main part. So first, let's... Yeah, do you want to discuss tissue architecture point in any way? Any questions? Okay. Cool. So now we get to the main point, and the main point is about how can we use single cell and spatial transcriptomics data to establish comprehensive spatial maps of tissues, and then analyze those maps in to either get at tissue architecture point or at the cell type collocation point. And this was the focus of our work that produced cell to location paper. A lot of people contributed to that paper, and this is the names of people that have made especially big contributions. I think they will be also named on slides where they're relevant. But yeah, I guess I want to highlight Artyom and Alexander Evazidis for the help in figuring out how to do the modeling of this data, and the rest of the people for help with various analysis, as you will see in the slides. And I will also make kind of general acknowledgements for the previous part of the talk later. Okay. So first thing that we need to discuss is how this spatial transcriptomics technology work. There is a number of technologies that are called spatial transcriptomics. Here in this section, I will only discuss spatial transcriptomics technologies that work by next generation sequencing. And Victor will later come in and talk about other types of technologies get at the same problem. In spatial sequencing technologies, what we have is what we have are arrays of specially barcoded capture nucleotides. What these nucleotides allow you to do is that they allow you to hybridize mRNA from cells and then do reverse transcription on this specially anchored primer. And the way you get mRNA from cells onto the special locations is by putting tissue directly on top. So most widely used technology, Tenax Visium, prints oligonucleotides on glass slides. So and different positions on the glass slide have different barcode in the sequence of this oligonucleotide, and these oligonucleotides are anchored to the position. So then on top of that glass slide, you just directly put the tissue. To that tissue, you apply certain enzymes to enable RNA dissociation, and then RNA can dissociate, diffuse towards the tissue. I don't remember all of the exact steps of the protocol. You can read them. Tenax generally has very good documentation of experimental stuff. You can read kind of how exactly it happens with pictures. The main point is that you detect RNA by directly capturing RNA at the spatially distinct positions. And there is a number of related technologies such as slide-seq and stereo-seq, which define positions differently rather than printing nucleotides in the way they are printed is somewhat similar to how you use inkjet printers on paper. You can use other ways to create these barcoded locations. But the main idea is that you directly capture RNA. As there is also now new technologies arising that use fixed tissue, and rather than detecting RNA, they detect barcodes. But the main point is that you detect some stuff from the tissue, and you then measure how much RNA you detected by doing next generation sequencing. And when you have next generation sequencing, you can look at the sequences of the barcodes, you can look at the sequence of reverse transcribed RNA, and you can decode which RNA was detected at which position. Hopefully that makes sense. One of the major limitations of these technologies is that they are not single cell. There has been a kind of dramatic improvement. This slide is a bit old, but actually the proof of concept papers, a lot of proof of concept papers came around 2019. However, actually commercializing technologies and making them happen in practice took quite a long time. So, for example, SlideSeq was published back then, but it's only now getting commercialized as Curia and offered to researchers for more easy use. And generally, until these technologies are commercialized, the only labs who can do them are the labs that developed them, or the people who worked in the labs that developed technology. Yeah, so the resolution has been improving. It somewhat reached theoretical maximum with SlideSeq and StereoSeq methods. However, even though there is, there are 2 points to make. One is that Visium spatial transcriptomics, as the most popular technology, is not single cell. What this means is not just that it captures multiple cells, is that it captures multiple tissue architectures in one. It captures RNA from cells of multiple tissue architectures. So, for example, the B cell zone with B cells plus blood vessels, capillary type blood vessel plus arterial type blood vessel. So you can have quite a lot of intermixing. But even those high resolution technologies, they don't have. It's not like you see where cells are as you do in microscopy. You just capture RNA from special locations, and you have to guess where the cells were. And the cells can non-perfectly overlap with these locations, such that some locations actually capture RNA from one cell, but many, about half based on our experience with SlideSeq, capture RNA from multiple adjacent cell types. So you still retain the same problem, but it now gets a lot more complex because you have a lot higher resolution, other issues to deal with. Yeah, we are moving in the high resolution direction, but the computational methods in that space haven't caught up to what you can do experimentally. We are mainly good at doing this. Do you have any questions about the technology?
So you have several cells in one dot, but then do you trying to compute what actually which cells actually they are, or how do you handle this problem?
That's the idea behind our method. So we will discuss this next. So we wanted to solve that problem, and on top of this, we had a bunch of criteria for what this model, for how we need to address this. First is that we needed to enable mapping of similar and rare cell types, transcriptionally similar and rare. Then we needed to achieve high sensitivity to enable rich downstream analysis of the types that we discussed, like cell communication, collocation, inferences. We also recognized early on that people want to use complex single cell references with multiple batches, multiple technologies, rather than just the simplest possible case that statisticians always like to solve. And we also wanted to formulate in a model in a way that allows us to get some future proofing, that establishes the base of how you solve this problem, that enables extension to these future technologies, the better technologies such as SlideSeq. or modifications in chemistry, such as using fixed tissues as opposed to RNA from fresh tissues. And with this in mind, we came up with the following model and the following problem formulation. So what we can, I will not go into the details of how we get reference signatures from an atlas, from a complex atlas with multiple batches and technologies. I will just say that what we need to start with is abundance of RNA of genes ABCD in cell types, and this is just average amount of RNA molecules corrected for those technology effects, batch effects. Given this information, given these reference signatures, what we want to find is abundance of cell types at these distinct spatial locations. And the data we have is abundance of RNA at these distinct spatial locations. So we need to put the two and two together. And the way we do this is by formulating a generative count model of the observed spatial counts. And we factorize expected spatial count into abundance of cell types by using reference signatures from the single cell atlas. However, to do that successfully, we need to account for one biological property of the tissues. The cell types are not randomly distributed and they collocate with each other. And we need to give a model an option to learn which cell types tend to collocate, to do that robustly. And on top of that, abundance is a positive value that depends on how much RNA is detected in the tissue. If you have 50 cells in one of these spots, in one of these locations, you will get different RNA abundance than if you have 5 cells or 1 cell. So we need to deal with this, and we deal with this by asking users to provide expected number of cells and then learning some values based on this input. And expected number of cells across the tissue, not for every location. So these are the main biological properties that the model needs to deal with. And then we have a bunch of technical properties. First, we need to account for the difference between single cell and spatial transcriptomics technology. You can think of it as a regressing the difference between them by using this multiplicative mg parameter. Yeah, G index, yeah, I should have explained the equations in a bit more detail, but I will do this once I name all of the concepts. We also need to do per location normalization, then account for additive background. So this would be the RNA that got captured, but not because it came from the cell on top of the location, but because it diffused there from somewhere else. And then finally, we also need to give the model some leeway in while we tried to explain what we know about technical factors, there are other possible differences, and we need to give the model an option to explain that unknown variance. So, yeah, I should have explained the equation. The idea is that we can factorize abundance of RNA for every location S and every gene G using this equation with these terms that we just discussed. And the core of the model is that we sum up over the contribution of cell types, where we just multiply abundance of cell types that we estimate from provided reference signatures. Then we scale that by technology difference, and then on top of that add the background terms for every experiment, for every section that you put on through this technology, for every tissue section that you put through this technology, and then you scale both the background and biological expression by per location normalization. And what this normalization deals with is gradients in how well detection works. And in some experiments it is not really necessary, but in practice we see that in most experiments, especially from more challenging tissues rather than perfectly preserved mouse tissues, you actually have to do quite a lot of normalization. And yeah, so hopefully that makes sense. Any questions about the model?
I ask a technical question. How do you measure the fit of the model that it works well? What are the criteria?
We look at number of QC plots that you will look at in the tutorial, but you are leaving, or maybe actually, what we want to know is that RNA abundance is explained well, so we can compare observed spatial counts to this average prediction from the model, and if there are, we found that it's generally enough to just plot scatter plot, a density plot that shows how much differences there are, and generally it's very highly correlated. I don't have a plot in my slide, so I can show, but it's shown in the in salt location tutorial. Generally, you have almost perfect correlation, and if you don't get that, then indeed there are issues. But overall, this approach seems to be relatively robust. Probably one of the best ways to break it is to provide bad reference signatures. Either incorrectly kind of normalize them, not estimate them according to our procedure, or let's say if we take a complex tissue like the brain, cerebral cortex with tens of cell types, and you just provide 3 cell types. That's kind of a very good way to break this model and not get anything useful.
I would like to ask about cell types, because cell type is some selection and not very well defined, basically. And you can, based on single cell RNA sequencing, you can find more or less cell types depending on your resolution and then on your knowledge. If you're working with differentiation trajectories, then you will have a continuum of cell types, and therefore you kind of have like, should you have more fine graded clusters, like particular stage, pretty small, or is it better to combine everything in one big cell type, which is pretty like rough?
Yeah, that's a very good point. So the way this method works is by spatially mapping transcriptionally distinct clusters. So what you want to formulate in the reference are transcriptionally distinct clusters, and I will show you one example of how you can do this systematically. That would help with gradient problem, for example. There isn't any good way to map gradients at the moment, at least not that I have seen, kind of to directly map a gradient as opposed to stages within the gradient. And what you want to do is use the reference clusters of maximum granularity. So not have a cluster of neurons, but have a cluster of inhibitory and excitatory neuron of layer 4, this subtype, excitatory layer of layer 4, that subtype. So you have to do as much granularity as you can afford in your research study, with some exceptions that we can talk about in the workshop part. So now I want to briefly show you some the most important points about comparison of our method to what was put out before, and some of the concerns that you can think about, and also how to formulate benchmarks in your work if you ever need to do that, and then show those 3 biological examples that I mentioned. So the first thing that you need to be concerned about is whether model correctly exclude cell types that should not be present in the reference. So let's say in this in the simulated example, it's not simulated, it's contrived example. We take special data of the visual of the somatosensory cortex, and we construct the reference using cell types of somatosensory cortex, but also hippocampal area, which is not present anywhere in the tissue. And the idea here is that we want to test if we give the model lots of extra cell types, does it deal with it correctly by saying that, well, they are not present anywhere in the tissue, or does it actually learn that they are present? So we expect top to map, bottom to be absent. And if we did a comparison of this for cell location as well as for other competing methods that existed at the time, and what we clearly saw is that cell location can distinguish them, maybe not perfectly, but there is kind of a large, but what we see is that mean proportion of these cell types mean proportion of the cell types that we expect to be present in the tissue is 5 times higher than that of cell types that we don't. So there is quite a lot of specificity, even though individual cell types may still be mapped, which shouldn't be. However, all other methods have much weaker specificity. And we suspect this is because we are using a Bayesian model with the priors that serve as a regularization on all parameters, whereas other models just directly estimate cell abundance values. And for some of the methods, the relationship was even reversed. And the method mapped cells that shouldn't be mapped and somewhat excluded stuff that did. Actually, no, it doesn't quite show this. But kind of methods struggle to do this, and even stereoscope, which is somewhat similar in architecture to ours, struggles with this as well. But we do well. So you can somewhat rely on the ability of the method to exclude cells that are not there. So then the second thing that we need to test is whether, I mentioned in the beginning that we wanted to have a method which allows you to have this high specificity for fine-grained, transcriptionally fine-grained cell types. Can we put this to the test? And we put this to the test by using human gut Visium, human colon Visium, and human colon single-cell atlas. This atlas represents all of the tissues within the gut, including epithelial, mesenchymal, immune cells. And what we expect is that most of the lymphoid immune cells should be present in this lymphoid structure in the spatial data. And because Visium is a technology paired with histology images, we can use the histology image to annotate where these lymphoid regions are. The first thing that we looked at is whether if we do spatial mapping of all of these cell types and we map them at the granularity shown in color, not in these outlines, we see cell types that define major tissue zones, especially the gradient of differentiation in the epithelial layer. And this means that overall the analysis worked, but now we want to systematically assess whether lymphoid immune cells indeed map to the lymphoid structure. On top of this, we need to formulate an objective way to define what is transcriptionally fine-grained. And we did that by comparing reference cell type signatures of these cell types using Euclidean distance, and by picking the distance to the most similar cell type. So let's take Tregs, we pick Tregs, then we look at the similarity to all of the other cell types and pick the value for the most similar cell type. Repeat this for all of the lymphoid cells, and we get a distribution of how transcriptionally distinct they are. And we just split them into distinct group and fine-grained group like this. And what we can now do is we can now test how specifically are the lymphoid cells mapped to this area and excluded from the rest of the tissue, and stratify the comparison by transcriptional distinctiveness. And what we see is that while other methods, especially RCTD, are quite good at mapping transcriptionally distinct cell types, we are especially good at mapping transcriptionally fine-grained stuff. And why is that the case? That's the case because we account for cell type collocation point. We enable the model to learn which cell types tend to collocate, and we see that if we delete that part of the model, we lose all of the performance difference on fine-grained populations. So yeah, hopefully this benchmark shows you that method we developed is robust, as well as helps think about how to validate these things. So now we can go to the most interesting part, the examples. Any questions about benchmarks?
Yeah, you said previously that you teach the model to learn that the cells can collocate.
Yeah.
How do you learn that?
We use hierarchical priors in Bayesian modeling context. We say that abundance of cell types for every location in every cell type can be factorized into non-negative factors, and these factors, you can think of those factors as representing tissue zones. So, and because it's a hierarchical prior, it influences abundance estimates, but it's not constrained. It's not that we are saying that cell abundance is fully low dimensional. It just helps the model learn cell abundance. And we don't really interpret this parameter, it's more like an auxiliary tool that improves accuracy. But yeah, the main idea is that you can use non-negative matrix factorization to learn informative priors. Cool. Yeah, we are getting close to the end. But also, hopefully, the current examples are the most interesting ones. So, as Diana asked, how do we formulate reference clusters for this analysis? And the example that I want to bring up is that I think that we can use spatial regionalization as evidence to better define cell populations. And to do this, we looked at astrocytes, because astrocytes are, well, they are interesting from that auxiliary point of view. They support neurons. And the question is whether they need to adapt different identities to support neurons of various regions. And to do this, we generated Visium data and single nucleus data from adjacent tissue sections. So we took thick 200 micron sections to generate single nucleus, thin 10 micron sections to generate Visium data, and did multiple rounds of this throughout the depth of the brain through the area that includes hippocampus. And this section includes a lot of brain areas. And one of the points that we also looked at in this analysis is just if we overlay expected locations of these Visium spots as well as their size on top of the tissue, how many nuclei do we observe in each of these spots? And you can see two things in here. One is that the number of nuclei varies quite a bit, even within the cortical area. It's somewhere here. But also that you have partial cells. You have partial overlap of some of the nuclei with these capture areas. And actually, one of the important points that I didn't mention before is that you section the way these sections are sectioned on cooled microtomes is not perfect. So the section thickness can vary between individual samples, between different people who process the tissue. So there is this additional layer. And an important point is that because this layer is just 10 micron thick, it will because of that, it will capture some cells partially. So it doesn't really make sense to, well, we can look at the histology image and count nuclei. It doesn't really make sense to assume that we captured RNA from discrete 6 cells, discrete 13 cells. We captured RNA from that area, but we captured it from 13 partial cells. And this is just one caveat that you shouldn't really interpret the abundance from either our method or pretty much anyone who analyzes this type of data as exactly cell numbers. But yeah, let's get back to the astrocyte point. So we have single cell reference, we have Visium data, we can annotate single cell reference in reasonable amount of granularity for neuronal populations in these two colors, oligodendrocyte populations, microglia. We tried to annotate them at reasonable level, but not super fine-grained. Whereas with astrocytes, what we did is we isolated astrocyte cluster, we integrated it using the established workflows. And we did high granular clustering of the astrocyte populations. If you see main difference between telencephalic and diencephalic astrocytes, telencephalic are cortex and subcortical areas, diencephalic are thalamus and hypothalamus. We aimed for a high number of transcriptionally distinct clusters. And if you subcluster already, if you subcluster cell types like astrocytes, and you get a lot of clusters, you don't really know if they are real. You can examine the markers, you can examine the marker genes, as you learned earlier in this workshop, but they wouldn't necessarily tell you what they mean. And what we wanted to do is to specially map all of these astrocytes together with the rest of the reference. So kind of what we always want to do is we want to map the entire tissue, plus maybe a zoom in on your cells of interest. So we map them specially. We get in the high-level overview of the tissue. We can see which cell types collocate in which broad area of the brain, including astrocytes. But crucially, what we can do is we can compare the locations of these astrocytes across all of the sections from 2 mice, and we can group the clusters that have identical locations. For example, these clusters here were found to have exactly the same location in the hypothalamus. And we went through this systematically and grouped cell types with identical locations and derived 10 subtypes that are both molecularly distinct, so they have different gene expression profiles, but also spatially distinct. And here is an example of 5 of these 10 subtypes. One of the most interesting ones that we found were thalamic habenular astrocytes, which were very rare in the atlas, just about 40 cells, and very rare in the spatial data, but we can map them precisely, and we can get some evidence that even though these are super rare, they are probably distinct enough and doing something for neurons in that area. Yeah, any questions about this example?
Do you know maybe how the astrocytes sort of, I mean, I understand that it's only single cell data.
Yes.
And do you know how they compare to the human, let's say, groups of similar markers? Human astrocytes are quite distinct from mouse astrocytes, so
Yeah.
just wondering to what extent it's translatable into that.
I mean, Bayraktar group is interested in this question. There are some constraints on what we can do in human. Cool thing about this dataset is that we isolated nuclei and did spatial data on all of these regions at once. So we have data with no batch effects across the entire, across all of the regions that you see in this section. However, in human, we have to dissect region by region. And in general, as far as I understand, there are differences. There are certainly differences between the broad regions, such as thalamus and hypothalamus, or cortex versus amygdala. What is a bit more questionable is whether there are differences within areas, such as within the cortex. There are certainly some differences within the cortex that are probably related to whether astrocytes are contacting neurons versus blood vessels. So that probably exists. Whether the same exact subtypes exist in both species, I don't know. It's possible, but it can be analyzed in similar ways. Okay. Yeah, so hopefully this example illustrates that you can use spatial information to help define subtypes, and as well as how to approach systematic mapping. So systematic mapping would differ. Actually, one second. So systematic mapping differs from non-systematic in the sense that you can annotate your cells first and then do mapping, and your annotation will be based on what you know about the tissue. Whereas systematic point is that you just isolate all transcriptional differences, you map them, and then you annotate.
In the next slide, did you actually find any differences in the expression between the thalamic astrocytes that were spread throughout the brain, essentially, and correct to the dark blue cluster that seems to be scattered all around, right?
Habenular astrocytes have much higher abundances in the correct area called habenula. They are somewhat scattered around here, and this could be mistakes in mapping, because we also did validation using single molecule fish that Victor will probably talk about later today, and we see that these astrocytes are indeed present in that area.
We try to check whether there's here, if you took just, the groups which are scattered throughout the brain, if they differ.
So the way the method works is that we define reference, we compute average abundance of RNA in the reference, and we then ask where are the cells with that average abundance present in the tissue. We don't have cells that correspond to this, exactly to this area. Like we can stratify habenula astrocytes into the ones that are here and the ones that are in the correct area. And if that information isn't in the reference. So the way you can interpret this as kind of errors in mapping, or maybe transcriptional similarity of these astrocytes to white matter. It's not how kind of this technology works. I mean, all of these astrocytes have certain marker genes, but they don't really mean a lot to me and also to Omer. I mean, some of them do, but not all, and it's quite hard to guess what these astrocytes are doing just by marker genes. But they are clearly transcriptionally distinct. They are present in different. They are also spatially distinct, so it's possible that they are doing something. We now need to figure out what exactly. So we are getting very close to the end. We started a bit late, so I will take a few more minutes. So now let's look at an example which is closer to the cell communication point that we discussed earlier. Can we map microenvironments? Can we identify collocation? And our initial approach wasn't as thoughtful as what I presented to you earlier. I will just explain what it is, and then we can discuss. So we tested whether we can recover tissue areas using a tissue with very different architecture. The mouse brain and human brain in general has very clearly distinct anatomical areas that you can't really argue with. However, in the lymph nodes, what we have is more of a gradient of cell abundances, and mainly, if you look at the tissue, you can define several tissue zones. And these are the main ones. Kind of T-cell zone with certain T-cell subtypes, germinal centers where a B-cell development happens, and B-cell follicle zone where B-cells normally sit in lymph nodes and wait for bad things like infection to happen to people. And to do this analysis, we used single-cell reference, which comes from a number of tissues, including lymph nodes, but also including tonsils. And why tonsils? Because tonsils, because normally it's quite hard to get human lymph nodes from people who have an ongoing infection. And if you don't have an ongoing infection, then you wouldn't have much of a germinal center developing B cells or corresponding T cell subtypes. But you can get that in tonsils. And tonsils are also often surgically removed. So you can get the tissue from human donors. And this work was done by Hamish Kim, and he has several interesting papers about that problem. So we use that reference. It's quite mismatched to the Visium data. We took Visium data from 10x, and this is exactly the data set that we will look at in the tutorial. And the goal in the tutorial would be to reproduce the results that we observe here. So once we have cell abundance, this is just abundance of 3 cell types, but once we have everything, we can factorize cell abundance using the negative matrix factorization to get at distinct cellular neighborhoods. And again, we have to deal with the fact that because of Visium resolution, in these highly cellularly dense tissues, it will capture not just distinct cell types, but also distinct the structures, like blood vessel versus more functional part of the tissue. And here I'm showing you an example of cell types that were grouped into the same factor. And the top factor corresponds to dark zone of the germinal center. You can see kind of towards the top of these highlighted areas. And the bottom part is light zone of the germinal center. It's more towards the bottom of this specific germinal center instances. And what this means is that by applying a negative matrix factorization to cell collocation estimated cell abundance, we can find which cell types have similar collocation profiles. and automatically group them. And this approach works better than doing Leiden clustering precisely because of those overlapping compartments. And if we tried Leiden clustering of locations based on cell abundance, we varied resolutions, but we could never separate these distinct known zones of the germinal center. And in these zones, B cells that are going through the process of their development necessary for their antigen response are doing different things depending on the cell type they interact with. So it's quite an important thing to recover, and you have to recover that using factorization approaches, not clustering. And then the final example that I just wanted to briefly mention before we conclude is, and I mentioned the link to the workshop part, our work with Ilo and Amanda. So kind of they are main authors on this paper. The idea is that what we found in this paper is that there are certain niches within the tissue that contain plasma B cells, and these plasma B cells are specifically situated in gland areas of the lung airway. And the way we did that is by taking histology images, manually annotating the gland areas and all of the other areas in the tissue, and then comparing the systematic annotation to where the cell types are. And this is kind of an example from the factorization analysis that we can try to recover the same, but the point here, I want to highlight here is that in addition to more automated analysis, as shown in this example, what we can do is histological annotation and then comparing spatial transcriptomics to the histological annotation. And there are now methods popping up that allow you to do this automatically. For example, work from Nadav Yayon in Teichmann group. If you look him up, I will post some links to this. And he's proposing some ways to automatically create annotation of the tissue. Okay, so hopefully the cell to location part of the talk showed you that we can combine single cell and spatial transcriptomics to comprehensively map complex tissues. And by comprehensive mapping, I mean that you aim to map all cell types in the tissue, or at least as many as possible, as opposed to just mapping some cell types of interest. That I didn't really show you this point, but we also analyzed that this method applies to slide-seq data as well as nanostring and as well as recent FFP modifications of the Visium. So the method is quite robust to many technologies. Also for high resolution, we need new methods. We can also deal with complex references that come from multiple tissues and that are not fully matched to, that don't come from exact same samples, don't come from adjacent sections, and that we can also get some insights on microenvironments. So in the tutorial part, what we will look at is mapping of the lymph node tissue, and on top of the factorization analysis, we will try to get at this image. Try to quantify abundances of signals around target cells. So yeah, with this, I want to end. Thank everyone for your attention and discuss any questions about this last part.
Here, take the references from an atlas or the scRNA-seq. Do you just download it directly from the atlas and use it, or do you do any additional integration for specific?
This depends on kind of goals of your research project. In this case, we used exact reference published by Hamish because it worked, it worked for the applications that we wanted to do. I find that in many cases, when you take someone else's atlas, you see that it doesn't have the you don't really agree with their cell annotations. You might also have your own data, so you want to combine. One of the things that is quite valuable, I think, is that you can have your own data generated in this setup, but maybe not as much data as you would want. So in this case, we did 8 sections. Out of them, 5 worked, and 5 sections had 40,000 cells. And maybe there is an atlas with 500,000 cells. So you may want to combine them to both help you annotate and find more rare subtypes in your 40,000 cells. But yeah, also to improve the annotations. So depending on what you want to study, if you need to adjust what you do. We are thinking about kind of pre-training these reference signature models for cell atlases in HCA portal. We haven't gotten to that yet because of these considerations, like how many people would really agree to use someone else's annotations.
Question, but do you really need to do the full transcriptomics for the cells, or maybe there's just a subset of genes which is able to distinguish, like the majority of cells between, you know, across the tissue slice?
So the question is, can we, I don't know, maybe save money, save costs by, can we measure a subset of genes to get exactly the same result? So the challenge is that, do you know that are you sure that you picked all of the relevant genes? Yes, and this is the reason to use unsupervised spatial transcriptomics. I mean, Victor will talk about the imaging-based methods where you have to select genes. Yeah, that's certainly a consideration. And one of the concerns with FFPE methods, so for example, the next released CytAssist technology, which in some extent works beautifully, they adjusted, the technology works by hybridizing oligonucleotides to RNA rather than detecting RNA directly. And because you hybridize detection oligonucleotides, you have to design them. And they have done I guess they have done reasonably good work designing those nucleotides, but you can't cover all. You already bias your set to certain gene models. And to some extent, the technology works beautifully because you can adjust the number of probes to guide the sensitivity. You don't want to profile too many ribosomal genes which are high, but you actually want more probes for rare genes. So if you do kind of targeted panels, you have that option, and that's nice. But the concern is that we don't know to what extent those probes are specific, and also it wouldn't be applicable to any kind of species, whereas this special transcriptomics technology can be applied to kind of unusual species like tortoises and lizards and so on. And I've seen some examples of that recently. So kind of that would be one of the positive sides of just profiling RNA directly. I mean, designing panels is very expensive. It's something like 15 to 20K. So if you have to modify the panel, you need to have sufficiently large project to do it. Whereas here you can just apply it. Cool. Well, coffee break time.